And good morning, good morning, good morning, good morning. It's Monday. I hope everybody's dealt with their, if you're affected or afflicted by this, with your post World Series grief, and that we can get underway with where we have been touring through the invertebrate world. Let me just check the... Yeah, Olivia, that's the same thing. Kevin was, there was a little bit of thought about naming him Pickle or something like that, but it was Dr. Jacobs that was like, this is a human name. So we have Doug the dog and Kevin the cat now. Alrighty. Where have we been? We have been thinking about sensory systems. We started out last Wednesday in the sensory kind of chapter of our time together, thinking about our response to light. Which is kind of omnipresent across, even if it's just shade and kind of phototaxis, we see it rampantly across the invertebrate taxa of the world, let alone where we get into image forming and kind of the amazingness of these compound eyes that you see underneath there. And where are we going? Well, today we're going to be talking about tasting and smelling, and we're going to jump across multiple phyla here, annelids and mollusks, but we're going to spend a lot of time with arthropods, thinking about antennas and sensillas and pits. Excuse me, I promised last week that there'd be lots with pits. This is the pits, it continues. Pits is how we concentrate things. So we'll talk about smelling and tasting. And then on Wednesday, we'll keep talking about hearing and touching. That's kind of the mechanoreceptors are on Wednesday. So my hopes for all of you today is that we can appraise, synthesize, and interpret different examples of the variety, like some, I'll say, amuse-bouche of the enormous number of examples and varieties that the invertebrates have for chemoreception to sense and respond to their environment, and in particular how they are used by predators and prey to navigate the world. So we talked about last week, this kind of little set of arrows in terms of orienting us with the modalities that we're talking about. So from our perspective in a common language we're talking about, so when I'm talking about to the kids this morning, to our boys about what we're talking about and sharing with them that the Simpsons would be in today's lecture, we talked about this is tasting and smelling. And I've talked about the modalities as being one of the lenses that I have for seeing the world. Most of the world is small. Most of the world has no name. Most of the world does not care so much about vision. It's a smeller and taster. This is a smelling and tasting kind of world that we live in. And when we talk about chemoreception, it's basically, so if you wanted like the most wide, the broadest definition, it's the sensing of chemicals in the environment in and around an organism. And our traditional way, and this is kind of one of our biases, and it dates back millennia to Aristotle. And remember, Aristotle got a lot of things right, but he also thought flies had more legs than six, and this is in kind of rejection of an abundance of evidence. But he did get this right, or we've used it a lot since then. It's endured. That we think about olfaction, smell, and gustation, or taste. And that division, though, really we're talking about pores and we're talking about chemical properties, the sensing of chemicals. And so I just wanted to call attention to the fact that division, that modality, that has more to do with us than it has to do with the world around us. So broadly speaking, again, before we get into the examples, the chemicals in the environment, the odorants or the tastants, the chemicals, are detected using different organs and organelles by olfactory receptor neurons and gustatory receptor neurons, ORNs or GRNs. And I think we could just call, if you wanted to, you could make an argument in your growing biological career just to call them CRNs, chemoreceptory, chemical receptory neurons. Those neurons are typically housed in sensilla. Now the sensilla, what I always went to the sensilla, these are little hairs, hair-like structures that contain chemosensory neurons surrounded by support cells. And then when they're connected to the nervous system of the organism in question, we have odorant receptors and gustatory receptors that mediate whether or not we're tasting or we're smelling. And then, because we're talking about this not as a binary, but more as an anthropogenically tinged continuum, we have ionotropic receptors that can do kind of either. They're just responding to chemicals. And this is a critical importance, right? Like, it's clear, I think if you were to defend to friends on the bus or at the breakfast table, why are we talking about this in this class about invertebrate diversity? Well, structure and function of the most diverse group of animals, this is going to be really important. And it has been important already when we've talked about feeding, when we've talked about social behaviors, mating behaviors, predatory or prey, escape, homing, living on a territory. All of those bring in to the game the fact they're mechanistically connected to chemoreception. So let's think about the sensia. This is a cross-section from an annual review article that you can see down here. But we've got through this kind of with these olfactory receptor neuron cell bodies at the bottom. And you see the strands kind of beautifully kind of winding up into and above and out into the environment. So extending out, even if it's micrometers, out into the environment, these pore tubules, these little openings of chitin, if it's an ant or a fly, that allow the external environment in to touch and bump into via the sensory lymph, the hemolymph that's kind of moving around inside the body. But the chemical substances are then absorbed and then tasted off of these dendrites, these neuronal dendrites. But those pores, how they're constructed, how densely they are, there's a number of different ways you could be more or less chemoreceptive. You can have more pores. You can have bigger or smaller pores that will kind of help you sort out what kind of chemicals you're sifting in from the environment. This is essentially a kind of a hockey stick of a siftate or of a sifter for the chemical environment. You can also change the number of dendrites, the number of olfactory receptors, just the genetic or the genomic complexity that underlies this. There's a whole bunch of different ways to alter this. You can change the number of sensilla per unit of the body, whether it's on your antenna or a segment of your antenna. But these are all out. So we talked about the sensilla. This is the cross-section. And here is some of them writ large in two different parts of tasting tips. On the lips, the palps of a staphylinid. We'll see pictures of staphylinids today. This is a bias or an enthusiasm of mine. These are the rove beetles, the ones that don't have the elytra that covers the whole body, but that leaves many of the abdominal segments exposed. I've mentioned their diversity before. There are currently, even considering the fact that most staphylinid species don't have a name yet, we have already named as many species of staphylinid as exist species of vertebrate. So this is an inordinately diverse group of an inordinately diverse group. But these are part of the palps inside. So underneath kind of the chopping mandibles, we have these sensing organisms. So you see SNS, and you see little pits containing these hairs or hairs that extend out on top of the body, where you can see lots and lots of receptor bundles and these sensilla. And if we zoomed in even more, you would start to see those pores inside of them. So, and bundles would be on this moth antenna here. So we have the importance of detecting chemical signals from the environment. We often kind of stumble blindly in terms of what signals are being produced and by whom and for whom. And that's just to do with our own bias. We are a visual thing that only secondarily tastes and smells. So we often have to think empathetically. We have to try and, as scientists, as sharers of the planet, of their world, we need to think from their perspective, how might these communications pass through the world? How do they pass through the air? How do they pass through the water? How does being in water modify those signals? How does the directionality, either of wind or of current, modify or direct those signals? And because that's incredibly important for understanding a huge suite of behavioral responses and a huge suite of ecological relationships or problems, from settlement to mating to feeding to host-parasite relationships. Okay. Now, before we jump into some of the structures and some of the examples from different taxa that we'll spend the meat of this of our shared time together with, Caramel's going to remind me to jump down, and I don't see any questions, just love for Kevin, which is great. All right. One of the first ones that we'll deal with are in annelids. Now, polychaete annelids and many other annelids. So maybe the third most diverse group. And so from your earthworms to your leeches to those homopterous, the beautiful swimming things. One of the synapomorphies for these groups that we've talked about way back into 2700 has been this sensory organ, this nuchal organ. And we've presumed, because of its so this is kind of the line diagram of a polychaete annelid. So the segments are kind of blurred out at the bottom here under my mouse, but we've got at the anterior end, we've got antenna, and we've got labial palp, so lots of sensilla, but we've got these deep pits right there and right there. And these are heavily innervated, all sorts of tight connection to these olfactory sensory neurons from 3 different organs that project. These are the glomular, the olfactory glomulus, which you can say with a kind of a Harry Potter-esque inflection if you want to make it a spell. But the olfactory glomulus and the connection, the direct connection via these olfactory sensory neurons at the base of the nuchal organ really suggests that these are heavily important or used for chemosensory reception. We've inferred that they're used for things that reading the water, important for larval settlement because they exist. This is an adult picture, but they exist as larvae in the larva. They're feeding, sheltering, reproduction, social and aggressive interactions. And feeding is important. There's a better diagram here from your text. You can see these almost like ears. This is them listening to the chemical environment. These ciliated sensory pits or slits. So they have tentacles that surround them, directing current and water into them and around them, kind of sampling the environment. Chemoreceptive organs that are heavily, as you saw, innervated and connected to the central nervous system. And while the diversity of them and their shape and form changes dramatically across larger taxa, like across families and orders, those kinds of things, we tend to see them. It is still, from a phylogenetic perspective, we consider them a synapomorphy. That's still there are some quarters of the phylogenetic community where that's still up for debate, whether or not this is a shared characteristic, a shared character that is seen across all annelids, or whether it's arisen multiple times, because a ciliated pit on the anterior end is just a great way of sampling the environment, and then we have the influences of convergent evolution. So whether this is a true homology, I would say 70% of the evidence seems to lean that way, but there's still some quarters of the internet where it's not entirely sure. So this is an even better. So we've gone from a kind of fragmented diagram to a better line diagram from your text to a gorgeous Muppet-like scanning electron microscope of these ciliated sensory pits. So these are not eyes, these are the nuchal organs. And we see experimental evidence that when they're removed, the organism doesn't feed. That's one of the biggest kind of inferences that we can make, or in support for the hypothesis that they're involved in feeding, is that when they are experimentally removed in some brutal physiology experiments, the worm doesn't feed. And we also see, from a more observational perspective, that we see the greatest diversity of size and form in predatory species like this, where we see this kind of convoluted crown of cilia and tentacles on the upper end of the head. Now, this association with form and function, so form, how the shape and the number of convolutions, the projection or the depth of the pit, the number of cilia, the number of tentacles around, all of those have to do with the function of being a chemoreceptor, but they also then secondarily have to do with, well, what are you tasting for? Are you tasting in this last bullet? Are you tasting because you're a predator and you're keeping an eye? See, there's again with our bias, our visual bias, to be aware of is to keep an eye on. To see, meaning I understand. This is inappropriate terminology for the smellers and the tasters of the world, the vast majority of things. So here's two leech species, Whitmania pigra and Hirudonipponia. There's the look at the, I'm going to mute, here comes a cough. I hope I caught that and it didn't disrupt your own mechanoreception. So in the A of each one of these images, you can see kind of the full end of the body. C is looking at the cincilla. Two different leech species. So same group, same larger taxon of annelid here. They are both leeches. But very different looking sensory cilia and pore structures that are related to reading the mucus that the leech is secreting and interacting with the environment to bring chemicals in. So here I've blown up the C panel from each of them, and you can see much more densely ciliated, much more heavily ciliated. This is kind of a 5 o'clock shadow on a Tom Selleck kind of a thing. There's a lot of cilia there. It's very hairy. Whereas down here, this is more of a teenage kind of a beard or my teenage mustache, my grade 10 class photo mustache down here on the bottom with Hirudonipponia. So different functions during foraging, and only one type of chemoreceptor on Nipponia, which is down here. So that form and function being associated with how they're using what they're reading from the environment. Now, if we jump taxa from the annelids over to spend some time with mollusks, there's a bunch of different ways that we've talked about with mollusks. This one that's imaged here is for the gastropods with the tentacles. So if we take a look, so we've got kind of the shell and we've got the head and the brain and we've got these two tentacles, and out at the tenticular ganglion, so a particularly dense set of nerves right at the tip of the tentacle that is innervated densely with olfactory sensory neurons in the epithelium, in the skin of the mollusk. And so there is photoreception. You can see the little disc of the eye, but it's a really densely packed sensory area, sensory tip that can do photoreception, and it's tasting the air as well. So we have the tenticular sensory neurons and the olfactory sensory neurons that are connected again to these glomuli and the extension of that ganglion that exists out there. So the importance somewhat inferred here, the importance to these signals and to dealing with these signals by the fact that we're not even going to waste fractions of a fraction of a second to send them back and deal with a lot of the reception and the interpretation in the brain. Of course, some of that happens, but we have thickenings and innervation of the nervous system out in place, in situ, so that the response can be near instantaneous. And that's response to what? Response to feeding, response to orientation needs, response to homing. And in addition, if we were to flip over this diagram, we would see sense organs in the foot. Now, sometimes when we think about particularly gastropods, but let's say like an ancestral type mollusk, we think about the radula, right? The rasping and the feeding, and well, you're just you're sniffing the world, but really you're just looking for your own particular brand of algae. But of course, many mollusks, of course many mollusks, but even many gastropods are not raspers. They're not herbivores. They are predators. And we see that response with a lot of the surfing snails. If you remember the surfing snails where their foot can go out into the current and carry them back and forth with the receding tide onto these, and so that they can feed on decomposing fish. And what we've got here is a researcher poking with the shell tip, but also adding a little bit of fish juice, let's call it a can of tuna juice there, and responding just as Kevin the cat would to that. It's sending out a feeding flap and boom. So that's really fast response, in this case likely because of the innervation not in any particular tentacle, as you saw in the stylized or the generalized last gastropod example, but here because of sensory chemoreceptors in the foot. And if we continue to revisit in your brain, going through old files, thinking about mollusks, we talked, I think you'll remember, we talked a lot about the importance of a synapomorphy for the mollusks called the Osphradium. And this was hanging in the mantle cavity or hanging in sometimes in association with gills, but sitting somewhere inside where water is coming into and across and out of as being a chemoreceptive organ. We talked about it in 2700 a lot. I held off talking about it until we were talking about the bivalves because this is kind of one of the most iconic places. But it appeared in our stylized ancestral mollusk diagram that we kind of flipped through as we spent many lectures last year with the mollusks. So it's the organ for the mollusks that is tasting the water, particularly with more sessile ones that are both respiring with gills but also feeding with their gills. So it can detect food particles, it can detect size, it can detect, because if you're sessile, you're generally also just releasing your gametes into the water, right? So it's being used to receive information from the water that might be the gametes of a conspecific that is like, oh, this is time. We've got to get materials out into the water so that we can have babies. And the size of the Osphradium, it can be microscopic or quite large. And in fact, we've talked about it kind of iconically as being, this is what it does. And we know in most bivalve groups, this is what it does. And if you were to go to a molluscan zoology conference, you would meet representatives from each of the groups that would probably say that based on the number of years that they have studied their group, they could probably tell you where the Osphradia is or how it's used. But in fact, even in this diagram down here is a diagram for just the polyplecopherans. So it's a polyplecopheran biologist speaking to other chitons, right? The other polyplecopheran biologist saying, these are examples of where we have considered where you have told us the polyplecopheran Osphradia is and how big it is and how important it is. The one here, this is so adjacent to the gill, these kind of gray lines, but not inside of it as it is in the upper diagram there. But this chemosensory organism, or organ rather, its function in many groups, particularly the not sessile and the more mobile you are, the more uncertain the role for the Osphradia is. And in fact, just like the nuchal organ, there is still some quarters of the phylogenetic community where whether or not this is a true synapomorphy for mollusks remains under debate. So there's still lots and lots of work to be done. Here's Caramel again to remind me, and I'm looking and I don't see any updated questions. We're going to switch from kind of an annelid, from a more water-based view of the world to a more terrestrial one. We'll think about chemoreception in terrestrial arthropods and here in insects. So we've got antenna, and we've got a brain, highly cephalized. Lots, as we talked about, the compound eyes, right, and the simple eyes, the median or the ocelli. Clearly, the importance of sight here is enormous. However, what we haven't yet had a chance to emphasize is the importance of the olfactory sensory neurons, that we see tons and tons. And you see the role, this kind of like, I don't know, like a roulette wheel of glomuli. So the olfactory system here is limited comparatively to the olfactory system we would see in the aquatic system, because we're really... it can really only test, it doesn't have the medium in which to kind of float around. They tend to fall out of the air more rapidly than they do in the water. So terrestrial insects really can detect chemicals that are nearby. But we see then greater emphasis, the importance of chemoreception you can infer from the fact that there is greater emphasis on different organs, like huge, gorgeous kind of antler type antenna, where the number of sensilla is just increased to the tens of thousands on each antennal, each of those bilaterally symmetrical antenna. So ants, as an example that we'll come to right at the end of class today, that can use their antenna to discriminate the cuticular hydrocarbons, so different chemicals that are likely to do with, from an abiotic perspective, with protection, with being protected against water, but water loss, but are also con-specific and sometimes even familial, like the same colony will have the same composition of cuticular hydrocarbons. And they use their antennas to kind of read that to say, oh, yes, you are the same species as me, and not only that, you're from the same colony. So we're just going to work together now. We're not going to fight. So if ants and Hymenoptera don't do it for you, if you're a moth and a butterfly person, well, then you're all about antenna, right? Especially if you're a moth person, because male moths, have famously feather-like antennae that are used to sense female pheromones. And we talked a little bit about this in some of the earlier case studies about males that were flying up the signal that the females are letting out from at a particular height, a particular time of day, and waiting for the males to kind of hunt down the source of that pheromone. And that they can do it even though transmission in the air isn't as efficient as it is in the water, because of the tens of thousands of sensilla that are arrayed on these feather-like antennae. They can detect these female sex pheromones over long distances. For the species in the top left, over 11 kilometers. And they're not producing great big bushels of this sex pheromone. It's like nanogram and picograms. And then the last bullet is thinking about the specific, like the flight behaviors that they're using to track these down, or the release behaviors that the choices that she's making in terms of where and when to release her important pico or nanograms. So this is a look in a hawk moth at a long sensillum. So it's so long it doesn't even fit on the diagram. But you can see the pores that open out. So this would be one little pore on one segment of one antenna that could be joined by 7, 10, 15,000 other ones. And you can see that the OD here, these are the dendrites that are kind of going up through the sensilla, the part of the hemolymph that's in the sensilla that's absorbing the chemical from the environment, and then coming down and binding on and feeding information into the olfactory receptor neurons that, in this case, for the selective natural selection through time, has fine-tuned the hawk moth to one particular chemical that we've named bombycil, which is part of the pheromone. It's part of the complement of chemicals, but a really descriptive part of the chemicals that the female is releasing. So pores in that cuticle and that sensilla let the pheromone enter the lymph and feed into that system and into the innervation of the organism and get them going. So 38% of those cuticular hairs are innervated towards bombycil of the 2,100 sensilla per section of, in this case, Manduca sexta. And they form brush-like arrays on each of the flagella, and so there's a 32% of them, of 3.6 times 10 to the 5 sensory neurons of the hawkmouth antenna are pheromone sensitive. So in terms of how the importance of tracking down sex pheromones, the importance of chemoreceptory processes or mechanisms underpinning kind of all, or maybe not all, but most sexual behavior in these organisms, in terrestrial arthropods, it can't be overstated. Now, without giving way entirely, we've left the marine environment, I'm pretty sure, but we'll kind of bring along some water-based ideas when we're thinking about arthropods, when we think about crustaceans. Because crustaceans famously have an organ that you'll see in lab, this aesthetic sensilla, which is, so at the lateral flagellum, the first antenna, so there's the lateral flagellum right there, and at the base of that, they have these aesthetic. And they have, again, enormous innervation and concentration of the nervous system that allows the chemical sensor, the senses for different chemicals from the water at the anterior end, but then they also have different sensory pathways that project out, particularly from the legs and associated with the legs and/or gills, the moving legs or the respiratory legs on their abdomen. Let's take a look at another line drawing here. So we've got there shown in blue the lateral flagellum, and the aesthetics here are here in blue. It's a little toothbrush inside of like guard sensilla. So there's sensilla that are doing not just the role of sensing the chemical environment, but also protecting some of the most sensitive, some of their more sensitive compatriots on this antenna. Now, these antennae, particularly at the anterior end of things like decapods or your crayfish and your lobster, they are bimodal from their modality. So they're both sensing and mechanoreceptors. So we have all these. So these guard sensilla are both mechano and chemoreceptors, but the aesthetics are largely chemoreceptors, and they're kind of gorgeous. And if you look at the feathered sensilla on an antennula of a crayfish, that's A and B here, these SEMs. They're beautiful. If you like feathers in birds, these are kind of gorgeous. And the base, you can see them kind of moving around, and you can see the importance of directionality here. If you looked at the base of the feathered sensilla, that they can be directed and oriented just as like a radar antenna or a TV antenna would need to be. You can look at the complexity of that base and infer that or understand that what that is about is the importance of being able to turn all of these innervated sensilla into the flow of that which you are trying to smell. Now, of course, from a crustacean perspective, we have many lineages. Most are marine. Many are aquatic. And then there's those that are marine, aquatic, and terrestrial, like one of my favorite, the isopods. And if you look across, within one lineage, across kind of a continuum of terrestrialization, you can infer the importance of the olfactory or the chemoreceptory role. So this is a marine isopod here that you're looking at. You're looking at the business end, little tiny eyes, and then you've got antennal segments here and here. So, and then if you blow up one of these segments, you can see how covered in sensilla they are. There's a whole kind of a hairbrush end or a walking bit of cilia there on the outside. So for a marine isopod, the olfactory pathway is a really important one. And think about also, many of these live in a deep abyssal zone. This is not, light's not super important here. This is smelling and tasting. And so we see a comparatively great investment in the chemoreceptory pathways. Whereas if you were to go down into your basement and kind of pick up something that was moist and look at the front end of the isopod that's greeting you down there, what you would see is a tiny, stubby antennule. So there's the comparative. So there's the first segment, and there's the first segment. So much more investment in the marine close relative in the marine lineage than in the terrestrial lineage. So the terrestrial taxa are characterized by shorter antennules with smaller aesthetics and less investment in the olfactory pathways. And then comparatively, even though this isn't like a dragonfly kind of investment in a compound eye, you can see there's much greater importance here in the eye if you look at comparatively the number of ommatidia or the amount of surface area that they take up. There's caramel. I see no. So we keep going, and we change taxa. So we're still thinking arthropods, we're still thinking terrestrial, and instead of thinking about, I guess we'll call a bias, so sex pheromone communication and antennual innervation and the concentration of sensilla on antella, particular on males, terrestrial things like moths. We study those things a lot. And there's a huge investment in how it's done, and lots of it has actually historically been provided by the U.S. military, which were really interested in how male moths kind of navigate the world and study and respond to minute signals that are diffusely coming into them from a complicated environment. But that's a bias, because there's a lot of arthropods that don't have antenna. How important is chemical communication to them. Like for something like spiders. Spiders don't have antenna. So we have here one kind of web weaver, Agriote burcheni, where the question was asked and answered, how do they perceive male sex pheromones? And the answer, it seems to be, is with their legs. Now, we mentioned in passing a couple of times where there was, yes, there's a tentacle in the mollusk, but it's also in the foot. And yes, there's an aesthetic in the crustacean and the isopod, but there's other receptors around the body. So here, they are in the spiders. They are in the legs. So we've got these little sensilla that are found in spider legs. These are hairy-legged spiders, minute. You can see the size, 5 micrometers. So if we take this cross-section here and we look, you can see breaks in that. These are the pores, and you can actually see the pores if you blow up. And these are called wall pore sensilla, and they're found in male spider legs. Here's a line diagram though. So there's the wall pores. You can see think about their importance as being related to the number of them. These are not a couple of different holes that are poked in to kind of very bluntly sample the world. This is, look at the number of wall pores there. You can see, I mean, it's kind of almost more pore than it is wall. Highly sensitive. And we see that female spiders don't have these, and young males don't have these sensilla, these wall pore sensilla. There's another kind of going out and looking at the hairy leg. You can see that we've got the wall pore sensilla in blue and a tip pore sensilla in red. Different kinds on the first walking leg, and in blue and in red you can see the distribution of where they are. And what the researchers have found is that contact and non-contact areas differ as to where they have wall pore sensilla. And the wall pore sensilla, just like the aesthetic was protected in that on the antennule of the crayfish by the guard sensilla, there's a bit of guarding here. We don't see, or we don't find wall pore sensilla contacting the surface. So here's a spider diagram with some interpretation by color of their legs. I'd like you to shake yourself awake and show me where you would expect to see most of the wall pore sensilla. Nice, nice. Yep. Thank you for the guess. Yes, thank you for the hypotheses. This is, in fact, this was part of the research. And so what they found was that the contact areas, you remember that, let me kind of get rid of these with appreciation, clear them off, and we'll go back to watching the video. So it's a slowed down video, but it's really specific. Like you see it's only the very tips of the legs that are touching. And so those are the contact areas. And it's in those contact areas that might get damaged because they are just like the soles of your shoes. They're going to get worn down. That's not where the wall pore sensilla are. In fact, they are in these infrequent, but more often in just completely in the non-contact areas. So they're not going to be damaged, and they're going to be, so the male spiders are sensing the female sex pheromones not just with their legs, but kind of with their thighs. That's where the sensory, so they're not being, it's not the distal part, it's not the working part. Now, because I have this enthusiasm for staphylinids, we'll move through two more of my enthusiasms, some staphylinid examples and some ant examples before we break up here. This is a gorgeous, large staphylinid that I find rarely in rainforests. And I want you to look at the front end of this body. Look at those jaws. These are the mandibles. This is like two different scythes that kind of reach out and probably grab, visually, again, look at the number of ommatidia. They reach out and grab things like millipedes and carry them back to consume them. So likely, in this case, a very visual hunter. And if we switch from a light to a scanning electron microscope, there you can see the orient, there's the great big jaw. And if we flip it over on its back and we look at its mouth, you can see lots of contact, kind of long sensilla that don't have a lot of hairs at the end. So it's not so much about tasting, it's more about touching and kind of helping to break apart and consume. This is a really visual predator. I'm going to switch to another taxon here that's found in some of the same forests. This is an Anodylus, so another species of staphylinid. But you see the mandibles here are these toothed, kind of 3 or 4 toothed, very blunt toothed, but this is a decomposer. This is a tasting and then tear apart and eat. So it's smelling around for decomposition and then tasting. And take a look at the difference in its mouth. It still has these long sensilla associated, like contact things associated with feeding efficiently. But look at these 2 areas right here. So a very different innervation of the mouth and a very different emphasis on chemoreception associated with a different feeding type. The first was predatory, and this is a decomposer. And if we take a look at the antennas of another, clear based on the number of ommatidia, this is a beetle that lives on the surface and is a mobile predator. But if we zoom in on its antenna, you can see these tiny little stumps. These are called, these are chemoreceptory devices. These are called little peg organs, chemosensory peg organs. This is a diagram of another one from a different arthropod from your text. But you can see if we could go back and take a cross-section of each one of these little stumps, that is hundreds of them on any one segment of any one antenna, so thousands across an organism, is just filled with different innervations and sensory bodies. That is thinking about intersecting the chemical environment. Now, it would be, I'll say it again, this is kind of a reoccurring theme if there's bingo going on. This could be a course in itself. If you wanted to think about the chemosensory world of the terrestrial world, ants is really who you should be studying. Ants in the kingdom of chemoreceptors, because to live socially as an ant means that you're living underground, so your visual sense is often limited or not important at all. And you're also living with hundreds to tens of thousands or to hundreds of thousands of your close relatives. And so all of the complex behaviors that ants use rely on the transmission and detection of chemical information. So things like the queen, how the queen interacts and receives information from the workers that are going up onto the surface once she has set up a colony, let alone the chemical information that she is giving to the world as she's calling males in on their nuptial flight. The one day when she's kind of released from the colony, meets the male, mates, goes underground, digs, and kind of raises her first colony by metabolizing her flight wings. Now, as an aside, that sexual encounter on her nuptial flight, she could then live for decades, and she's maintaining sperm from that encounter for those decades. How she does that, we don't yet know. If you wanted to be a very wealthy myrmecologist, you could figure that out. But there's also care that's given. So brood nurses, so this white circle, these are workers that are caring for developing larvae. There's ones in the blue circle that are making midden piles. You see those are the ones that are carrying little bits of garbage out and making the little kind of volcano of the laziest of the sidewalk ants. There's certainly food collection and aphid tending and colony defending. All of these things require a really advanced and a highly invested in chemoreceptory network. We'll do it comparatively here. We'll take a split, this dotted line right in the center. On the left-hand side, this is an ant brain and influencing or kind of demonstrating the importance of chemical reception, chemoreceptory, versus the fly brain on the right-hand side, which is a very visual thing and comparatively has much less investment and complexity in its chemoreceptory apparatus. So we see here in ants that there are odor receptory genes, or genes involved in dealing with the reception of chemical signals, 400 plus compared to 60. So an order of magnitude more genomic infrastructure. Orders of magnitude, again, more than 7,000 sensilla per antennal flagellum in the ant versus about 400 for a much reduced size of antenna for the fly. The sensilla can contain maybe hundreds of different, and that's this innervation here of olfactory sensory neurons. This was very apparent to our youngest Daniel when he saw this picture this morning. Like, which one of these has more investment in smelling? And it's like the ant, orders of magnitude more. And this is then related to complex behavior. So things you might have seen before. Ants, if you've been at just kind of lying on your stomach in the sun in the summertime looking at ants moving around, you've probably seen them using trail pheromones. So ant workers out randomly interacting with their environment, looking for food. They find some. So she returns to the nest, and as she's doing so, both with all sorts of glands. There's Duffer glands, there's poison glands. Ants are glandular. There's a lot of glands because of the importance of the chemoreceptory system. But they also, even in the tarsals, in the bottom of their feet, they have different tarsal compounds that they're releasing and laying a trail. So as it goes and it finds the nice bit of honey that you've laid onto the ground, it's going back to the nest and it's laying a trail that inside the nest, once that's communicated with antennal brushing, again, chemical communication, the workers go back out and begin to fill up. So as they go back and forth, they're essentially creating a pheromone or a chemical superhighway. And the thickness of that highway is related to the number of ants, but that's then related to the quality and the amount of food that is there. So they reinforce and they expand. And then once it's exploited, the food they're no longer reinforcing the trail. So the food is consumed and hungry workers kind of break, and so the trail is broken, and then ants go around to randomly searching for food. So that's like a trail pheromone. There's another thing that you might have bumped into with ants that I found fascinating, and that's tandem running. So that's going out where the scout lays a trail to the nest after finding food. This is the scout, this empty ant here. It recruits another worker, so it bumps into a conspecific. They tell each other. She speaks to her and says, with antennal brushing, and they can kind of read the cuticular hydrocarbons of their skeleton, of their cuticle, and understand that they are from the same colony. And the second begins to follow the first back to the food source. And then they both lay order trails as they return, and each returning ant can bring another worker, and then you've got this kind of exponential explosion. So the importance of chemicals and structures like sensilla, and even sensilla built on antenna, but then also the receptivity of different parts of the body that lead to this kind of blossoming of complexity that we see in the invertebrate world. But then the antenna is not all about chemoreception, and as a bridge to Wednesday, we'll talk about these brome bristles. These are these very stiff things that here are on legs and here are at antennal segments, so they can they're at the hinge point of that antennal segment. And these are proprioreceptors that are present in the antenna of all arthropods, and they're mechanoreceptors. So they're evolutionary homologs of leg plates, and they can mediate the antennal positioning. So remember we talked about with the aesthetic how important it was to face where. It's these brome bristles that are helping the organism do that. And my last kind of view from the staphylinids of the Costa Rican rain and cloud forest is we take a look at this high elevation rainforest, low elevation cloud forest staphylinid, and we zoom in on her antenna, and you see this first segment here. That's a brome bristle right there. And these are all sensilla that are smelling and tasting. So proprioceptors orienting, smelling, and tasting. God, there's so much more we could talk about. But you have other things to do, and Caramel's asleep. So this is where we've been. I've enjoyed it. I hope you have too. And where are we going? Two things to talk about this week. One of them is that it is, if you haven't checked out CourseLink already, Shayla has come down with something. So the labs this week are postponed and pushed to next week, where you'll talk about sensing and responding as we have been in the lecture. We'll finish our sensory chapter by thinking about mechanoreception on Wednesday. The case study, Thursday to Monday, of course. And this Friday, please don't lose track of it, is the deadline for your second InvertR experiment. And we'll talk about staticists. So, on Wednesday. So basically, take care of yourself until then. Take care of others around you. I'm going to stop recording. If there's any questions, you can hang around to unmute and ask them, and I'll see you Wednesday.